Okay, so previously we have, gone through the components of the mammalian transport system. We have already looked at the types of blood vessels, we have already looked at the types of blood cells, and we have already looked at the formation of the tissue fluid or the interstitial fluid. So today we are going to look at the function of hemoglobin in the mammalian transport system. So you should have known by now that hemoglobin plays a very important role in the cardiovascular system, okay, which is to transport oxygen from the lung to the rest part of the human body. Okay, and this is because your body cells need a constant supply of oxygen in order to be able to carry out aerobic respiration to produce those ATP, okay, that is needed by the body cells. Okay, and oxygen that is carried from the lung, okay, is transported around the body inside red blood cells when they combine with a protein inside the RBC known as hemoglobin. And we have learned about the structure of hemoglobin in chapter 2, right? So you should know by now that hemoglobin is a type of protein molecule, okay, that is made up of 4 polypeptide chains, right? 2 alpha chains and 2 beta chains. And in each of the polypeptide chain, okay, there will be a heme group, okay, that is located inside of them. So since you have 4 heme group in one hemoglobin molecule, so each hemoglobin would then be able to transport 4 oxygen molecule at a time. Okay, and the equation is shown here, right? Okay, so something new that you're going to learn in this chapter is about the hemoglobin dissociation curve. Because hemoglobin is a molecule that has a function to transport oxygen from one part of the body to another part of the body. So this hemoglobin molecule must be able to uh, pick up oxygen, okay, efficiently, and also it must be able to release oxygen, okay, when it reaches the respiring tissues. Okay, and hemoglobin has been shown to do this task perfectly. Okay, so there is an investigation that has been carried out to see how the hemoglobin behave. Okay, and what has been done is that the hemoglobin uh, are extracted from blood and exposed to a different concentration of oxygen. Okay, and this different concentration of oxygen is uh, shown as partial pressure of oxygen, as you can see in the x-axis here, right? So you start from 0 up to 14 kilopascal. Right? The unit is in kPa or kilopascal. Uh, the maximum amount of hemoglobin is measured, okay, depending on the different concentration of oxygen that has been subjected to it. Okay. And as you can see in this particular graph, okay, you will see that at low partial pressure of oxygen, the percentage of saturation of hemoglobin with oxygen is very low. Okay, for example, you can see at 2 kilopascal, okay, roughly about, okay, only 20% of the hemoglobin is actually, saturated with the oxygen molecule, right? But as the concentration gets higher, okay, you will see that more and more hemoglobin will be able to bind with the oxygen molecules, right? And if you look at the x-axis at 14 kilopascal, okay, roughly there will be about 98% of the hemoglobin, okay, will be able to combine with the oxygen. Okay, will be saturated with oxygen. Okay, so the general idea of the investigation is very simple, right? At lower concentration of oxygen or at lower partial pressure of oxygen, uh, less percentage of hemoglobin will combine with those oxygen. Okay, as the concentration goes up, so will the percentage of hemoglobin that will combine themselves with the oxygen. So it's a proportional kind of correlation, right? Lower partial pressure, lower percentage of saturation, higher partial pressure, higher percentage of saturation of hemoglobin with the oxygen. Okay, but one question that comes out from this investigation is, since it is a proportional correlation, why isn't the line or the graph that you get is a straight one, right? Why must you have an S-shaped kind of curve from the investigation that has been carried out? Okay, so there is a topic on the syllabus on your textbook that talks about this. Okay, the S-shaped curve of the investigation. Okay, so we're going to discuss about this in detail. Okay, like I said previously, when we learn chapter 2, okay, when we learn about the structure of the hemoglobin, okay, we know that hemoglobin is made up of four polypeptides, right? 2 alpha chains and 2 beta chains, and each of the polypeptide chains, okay, you will have one heme group or one iron group, okay, which will combine with the oxygen. Okay, and when we talk about this process, when we talk about the combination of oxygen with hemoglobin, okay, what happened is that when an oxygen molecule combines with the first heme group, right? Because the whole combination of oxygen with hemoglobin, okay, they don't really occurs simultaneously, right? Oxygen will combine with each heme group, okay, in the hemoglobin molecule one at a time. Okay, so when the first oxygen molecule move into the first heme group of the first polypeptide, okay, it will cause the whole hemoglobin molecule to be distorted slightly. Okay, so I repeat it again. Okay, you have 4 different polypeptide, therefore you have 4 different heme group. Okay, and each heme group will be able to combine themselves with one oxygen molecule, right? So when the first oxygen molecule move into the first heme group in the first polypeptide, it will cause the whole hemoglobin shape to be slightly distorted. Okay, and this in turn, okay, the distortion of the whole hemoglobin structure will make it easier for a second oxygen molecule to combine with the second heme group in the second polypeptide. Okay, and then, okay, this will also make it easier for the third oxygen molecule to combine with the third heme group in the third polypeptide. Okay, and finally, the movement of the third oxygen molecule in the third heme group will also make it easier for the fourth oxygen molecule, okay, to combine with the fourth heme group in the fourth polypeptide. Okay, so the shape of the hemoglobin dissociation curve reflects the way the oxygen atoms combine with hemoglobin molecules. If you look at the graph, okay, up to an oxygen partial pressure of around 2 kilopascal, on average, only one oxygen molecule is combined with each hemoglobin molecule. Once this oxygen molecule is combined, okay, it becomes successfully easier for the second and third oxygen molecule to combine. So the curve rises very steeply, right? Over this part of the curve, a small change in the partial pressure of oxygen causes a very large change in the amount of oxygen which is carried by the hemoglobin. So if you look at the S-shaped curve again, you will notice, or you will see that it correlates perfectly with what is going on in the human body in terms of oxygen transport, right? So at high partial pressure of oxygen, okay, which is usually takes place in the lung, you will see that, okay, the saturation, or the percentage saturation of hemoglobin with oxygen is very high, right? So this represents perfectly with what is going on inside the lung. Okay, high oxygen concentration when you breathe in, therefore high percentage of hemoglobin, okay, that will bind with those oxygen molecules. Okay, and if you go to the left of the graph, okay, at low partial pressure of oxygen, okay, which represents roughly what's going on in the respiring tissue, right? Because respiring tissue produces a lot of CO2 and they use up a lot of oxygen molecule. So at this part of the graph, it represents the partial pressure of oxygen at the respiring tissues. So at low partial pressure of oxygen, you will see that uh, the percentage saturation of hemoglobin with oxygen is also low. Why? Because hemoglobin will, release those oxygen that they are carrying to the respiring tissue for them to, carry out the aerobic respiration, right? So the whole thing correlates perfectly with the scenario that is going on in the lung as well as in the respiring tissues, okay, of the human body. So we have learned about the S-shaped curve, and we have basically know that the percentage saturation of hemoglobin is directly proportional with the concentration of oxygen, or the partial pressure of oxygen. Okay, but the partial pressure of oxygen alone, okay, is not responsible in determining how many hemoglobin can actually bind with the oxygen molecule, because the other factor that plays or that comes into the picture is the concentration of carbon dioxide, right? So not only partial pressure of oxygen, partial pressure of carbon dioxide will also plays a very important role in determining how many hemoglobin can actually bind with the oxygen molecules. Okay, and we have some terms that is used to describe that. Okay, and that term is known as the Bohr shift. So what is Bohr shift? Okay, let's look at this particular diagram first. Okay, in respiring tissue, what happened is that they produce a lot of carbon dioxide, right? And those carbon dioxide, okay, will be removed by the cells, okay, and they will go into the blood vessels. They will bind with the water in the plasma, okay, within the blood vessels to form what we call as carbonic acid. Okay, and this carbonic acid can dissociate themselves, okay, to form hydrogen ion and hydrogen carbonate ion. Okay, and the thing with these hydrogen ions, okay, that is formed by the dissociation of the carbonic acid, is that this hydrogen ion can bind readily with hemoglobin molecule, okay, in the RBC. Okay, so when they bind with the hemoglobin molecule in the RBC, the hemoglobin molecule will be forced to release the oxygen that they are carrying. Okay, so okay, when you look back at the S-shaped curve graph, okay, when you look back at the S-shaped curve graph, okay, whenever there is a high concentration of carbon dioxide, okay, theoretically what happens is that the hemoglobin will be forced to release the oxygen, okay, and therefore there will be less or there will be much less hemoglobin that is now concentrated with the oxygen molecules. Okay, and if you look at the graph, okay, at high partial pressure of CO2, okay, there is a tendency that the graph will be moved to the right. Okay, there will be a tendency for the graph to be moved to the right. Why? Because less oxygen is now combined with the hemoglobin, because some of it has been released, right, by the high concentration of carbon dioxide. Okay, and this is what we call as Bohr shift, because the high CO2 concentration will shift the graph to the right. Okay. So basically, hemoglobin will mop up the excess hydrogen, okay, that are formed when CO2 dissolve in water and dissociate, right? Because we don't want the hydrogen ions to accumulate, because high concentration of hydrogen ion, right? High concentration of hydrogen ion will reduce the pH of the plasma. And if the hydrogen ions were left in the plasma for a long period of time, okay, the blood will become very acidic. So in a way, this will help to maintain the pH of the blood. Okay, and the presence of high partial pressure of CO2 causes hemoglobin to release oxygen. Okay, and like I said, this is what we call as the Bohr effect, right? And if you look at the Bohr effect and relate it to the S-shaped curve, you will see that the Bohr effect will tend to move the curve to the right. Okay, so let's look. So now let's look at the transport of carbon dioxide, right? Uh, because in oxygen transport, basically it's very simple. Oxygen combined with hemoglobin to form oxyhemoglobin. Okay, from the lung, it is then transported to the respiring tissues, where oxyhemoglobin, dissociate to become oxygen and hemoglobin again. In carbon dioxide, however, there are a few different ways where this carbon dioxide can be transported from the respiring tissues, okay, into the blood. Okay, and I think the diagram here, it tells, or it sums up everything that you need to know. Okay, so if you look at this diagram, you can see that, okay, some of the carbon dioxide, okay, that has been released by the respiring cells will move into the blood vessel and then goes into the RBC, where it combines with the water molecules, right? So CO2 combines with water molecule, and with the help of an enzyme known as carbonic anhydrase, okay, it will form what we call as the carbonic acid. Okay, and carbonic acid, okay, will usually dissociate itself to form hydrogen ions and also hydrogen carbonate ions. Okay, and this is how most of the carbon dioxide is transported back to the lung to be uh, exhaled. Right, 85%, okay, of the carbon dioxide will travel back to the lung, okay, in this particular form. Okay, and then there is also some of the hemoglobin, okay, that will move into the RBC, combine with hemoglobin to form carbaminohemoglobin. Okay, and about 10% of carbon dioxide will be transported back to the lung in this particular formation. Okay, and then you have about 5% of carbon dioxide that will just move into the blood plasma, dissolve in the blood plasma as it is, right? So it will dissolve as carbon dioxide before it is being transported back to the lung. So 3 different ways in which carbon dioxide is transported to the lung. Remember, first is by the formation of hydrogen carbonate and hydrogen ion. Second is by the formation of carbaminohemoglobin. And the third one is, by dissolving directly in the blood plasma. Okay, let's look at some of the problems that might arise in oxygen transport. Okay, I think you have 2 listed in your syllabus, right? The first one is the presence of carbon monoxide. What is carbon monoxide? Carbon monoxide is a compound that is formed when a carbon-containing compound burns incompletely, right? And the thing with carbon monoxide that it combines with the heme group inside the hemoglobin to form carboxyhemoglobin. Okay, but carbon monoxide, okay, combines with hemoglobin 250 times more readily, more efficiently than it does with oxygen. So when you have carbon monoxide around, the person with those kind of situation will eventually die. Will eventually die by asphyxiation, right? Because I mean that person will be deprived of oxygen inside his body.